Hi there. I wanted to prepare another video today talking about transport of molecules across cell membranes. So this is a really important process for cells maintaining homeostasis, and there's a lot of complex detail here. So I thought we'd walk through it together and see if we can help understand. So we've been learning about cells, and one of the important things about cells is that they have to maintain constant internal conditions. The beginning of our biology class, we would have referred to that as homeostasis, right? Maintaining constant conditions. So here's a picture of a cell, right? It's got its cytoplasm inside, and then the cell membrane surrounds that cytoplasm, and then we've got the outside world. And there's a number of molecules and substances that we have to get across the membrane in order to maintain that homeostasis. So examples include things like nutrients. We're going to need to get things like glucose into the cell, so the cell can use it for energy. As the cell uses it for energy, we're going to create some waste products. We're going to need to get waste products out of the cell. Along the way, we're going to talk a lot about the use of oxygen and why carbon dioxide is produced. So these gases are going to have to get in and out of the cell. And then a lot of cells require some ions to work correctly, things like sodium and potassium, all sorts of other ions. We're going to have to get those in and out of the cell as well. So all of these things have to cross over the membrane, and we have to move them across the membrane a lot to make the cell work correctly. So it's really important that we understand the processes by which we can get these things across the membrane. So just as a reminder, the cell membrane is a really complex structure. We've talked about this phospholipid bilayer before. So there's the two sheets of phospholipids laying side by side. And there in the middle, we've got all those hydrophobic fatty acid tails. And on the face of the membrane on either side, we've got those hydrophilic phosphate head groups. Okay? But that's not the only part of the membrane. The membrane also includes all these additional proteins. And in this video, we'll be talking about what some of those proteins do to help us get things across the membrane. Okay. So we're going to talk about two major categories of moving things across membranes or transporting things across membranes. We're going to categorize all kinds of transport as either active or passive transport. So let's define what active and passive transport are before we move on and talk about the specific kinds. So I'm going to divide my membrane here in two halves, and we'll put active on one half and passive on the other. Again, here's our membrane with the phospholipid bilayer, with the hydrophobic interior and the hydrophilic head groups pointing out. And we're going to talk about passive transport. I'll do that on the left side of this line. And then we'll talk about active transport. I'll put some details on the right side of this line. So these are the two ways which we can get things across the membrane. And they both have to do, or they both are categorized by how a molecule is moving in relation to where its concentration is greater. So for passive transport, I'm going to draw in some molecules here. They're star-shaped molecules. I'm going to use these throughout today's video. So here on the top of the membrane, there's a high concentration of these stars. And at the bottom, there's a very low concentration of these stars. And we know from diffusion that any molecule wants to move or will freely move from where its concentration is high to where its concentration is low. And that's what passive transport is. Passive transport is just any time a molecule is moving from where its concentration is high to where its concentration is low. And it's going to cross a membrane along the way. Okay, so passive transport is moving down the concentration gradient from high to low. And the key detail about passive transport is that it does not require energy. Okay. So how about active transport? So same thing. We're going to be moving a molecule from one concentration to another. So I'm using stars again, high concentration above the membrane, low concentration below the membrane. In active transport, we're going to move the opposite direction. We're going to move a molecule from where its concentration is low to where its concentration is high. We would refer to this as moving the molecule against its concentration gradient. And the key detail about active transport is that this is going to require energy. Okay? So we're going to walk through a bunch of specific kinds of passive transport, and then we'll look at some specific examples of active transport. But the key delineation here is the energy requirement and which direction the molecule is moving in relation to its concentration. Okay, so let's look at passive transport first. We're going to think about three kinds of passive transport: simple diffusion, osmosis, and facilitated diffusion. Now there's going to be two kinds of facilitated diffusion, one that involves a channel protein and the other that involves a carrier protein. So we'll discuss those specifics when we get there as well. So let's start with simple diffusion. So simple diffusion is, again, a kind of passive transport. Simple diffusion works on a certain kind of molecule. It works on small and non-polar molecules. Small and non-polar molecules can fit through the gaps between phospholipids to cross the membrane. So one of these stars is going to shimmy its way between the phospholipids, between the fatty acid tails, and then through to the other side of the membrane, moving from where its concentration is high to where its concentration is low. That makes it passive transport. And simple diffusion, because it just simply diffuses right through the membrane. So here's what's going to happen, right? That star is going to move right through the membrane, through the gaps and spaces in between the phospholipids, and that's simple diffusion. Some examples of small and non-polar molecules that undergo simple diffusion through the membrane are things like oxygen gas and carbon dioxide gas. And those things are constantly crossing our cell membranes via simple diffusion. All right, so how about the next kind of passive transport? So let's think about osmosis. So osmosis is another kind of passive transport. Osmosis is when a water molecule moves across the membrane from an area of high water concentration to an area of low water concentration. So that sounds just like simple diffusion, doesn't it? The only difference is that it's water. And since water is so important to life, and we spend a lot of time thinking about water in biology, we've given the simple diffusion of water a special name. We call it osmosis. So it's the exact same process we just looked at. One of these stars, which is now going to represent a water molecule, is going to move across the membrane by squeezing in between the spaces and getting to the other side. And that's osmosis. Again, the only example of osmosis is water, because osmosis is the simple diffusion of water. Okay, so then we'll go to the last kind of passive transport, which is facilitated diffusion. So facilitated diffusion is going to be broken into two kinds, and we're going to talk about through a carrier protein first, but we'll get to that soon. So facilitated diffusion in some cases happens because the molecule that's moving is polar. So the thing about polar molecules is that they can't make their way through this layer of hydrophobic tails in the cell membrane. So remember that middle part of the cell membrane, the middle part of this sandwich, is all hydrophobic fatty acid tails. And hydrophobic things do not like to interact with polar things, and vice versa. So if one of these stars is polar, it's charged, and it tries to move through the membrane, it just bounces right off, because again, it can't get through these fatty acid tails. So in order to get one of these polar molecules through the membrane, we're going to have to provide some help. So another way to say helping is to facilitate. Right, so we're going to facilitate the diffusion of these molecules using a protein. And in this case, we're going to use a channel protein. So we're going to put a channel protein into the membrane. So it's this cylinder-shaped protein. It sits right in the membrane and it spans the lipid bilayer. And this channel protein has a little channel running through the middle of it. This black tube is a little channel running through the middle of the protein. Think of this protein like a straw, almost. So we just jammed the straw through the membrane. And now our polar molecule can go right through that straw. It doesn't have to interact with the fatty acid tails anymore. It gets to interact with this protein instead. And now our molecule has moved to the other side. So some examples of small polar things that transfer through these channel proteins are things like ions, things like sodium and potassium. Now there's a problem with this channel protein is we can't make that tube too big. Because if we made the tube really big, all sorts of smaller things would sneak through along with the big molecules that we wanted to let through. So that's why we have two kinds of facilitated diffusion. So this first one was through a channel protein. But if a molecule is large, they can't fit through the gaps between the phospholipids, and we can't let them through a channel protein either, because the hole in the channel protein would be far too large. Lots of small things would get through as well. So again, one of these stars, we'll consider one of these stars trying to get from where its concentration is high to where it's low. But this one's really big. So again, it's big, it bounces off the cell membrane because it can't squeeze between the phospholipids. So now we're going to we're going to facilitate the diffusion of this molecule with another protein. This one's called a carrier protein. So I'm going to put a carrier protein in the membrane. This one's shaped a little bit like Pac-Man. And when it's in the membrane, it's going to grab on to one of these molecules that needs to move across the membrane. Once it's grabbed on, it's going to close around it, and then it's going to open up to the other side of the membrane and let the molecule in. You see, by doing this process, we never have an open tube through the membrane that, again, lots of small things can fit through. So carrier proteins grab onto a large molecule, close around it, and then dump it onto the other side of the membrane. So some examples of molecules that are moved in this fashion would be things like glucose, amino acids, and nucleotides. So the monomers that we make the biological macromolecules out of are things that are commonly moved by facilitated diffusion. All right. So those were all simple, passive transport mechanisms. Next, we want to talk about active transport. And I think to save some time, I'm actually going to put active transport in a second video and post it right after this one so that the information is in bite-sized pieces. So in the next video, we'll talk about active transport, where we're going to think about pump proteins, bulk transport, which comes in two kinds, exocytosis and endocytosis. So look for that video if you're interested. As always, ask me questions if you have them on the discussion board, and I will talk to you soon.